the amino acids are the building blocks of peptides and proteins, whereas the nucleic acids are the ones which contain information about the proteins. The deoxyribonucleic acid, that is DNA, contains the blueprint of the protein in terms of the genetic code, whereas another nucleic acid, that is RNA, is the one which participates in translating that information from the DNA to the formation of protein in the cell. So in this whole thing, the proteins are the core of it. That's a central theme over here. And just taking another perspective, we know proteins as a dietary source, because it's a very important component of our diet, and because that is required in our diet, because that is very essential for the normal functioning of the body, because proteins play enormous amount of functions in the body. There are a lot of biological functions which they perform. Now, this whole series will be built up as follows. That is, to begin with, we will start our discussion on amino acids, and we'll focus, we'll spend a lot of time on understanding different dimensions, different aspects of amino acids. What are they, how do we represent them, and then how do we synthesize them, and then many more things about them. Thereafter, we will see that how do we move from amino acids to peptides, and then to proteins. We will talk about the structural and functional part of it to some extent there, and then towards the end we will give a brief account of the structural aspect of the nucleic acids. So that is how we are going to talk about in this whole series there. As regards today's session goes, today's session is going to be focusing primarily on amino acids. We will start about the session by talking about amino acids in terms of what are they, what is their structure, and then how do we represent them. And also we will talk about the nomenclature, the traditional nomenclature of alpha amino acids, that means on what basis have the names been given to them. Then as regards the representation of alpha amino acids go, we will talk about different kind of symbols given to them, the 3-letter symbols and 1-letter symbol, and so on and so forth. And then we will spend a lot of time on classification of alpha amino acids. We will talk about different bases of classifying amino acids, and then see how do we classify them, and then what is the significance of such a classification. And towards the end, we will try to sum up what we do in today's session. Okay. Now, let us see what are amino acids. We have just seen, we just mentioned earlier that amino acids basically are the building blocks of the proteins, but that is one bit of it. But if you really ask me, the number of amino acids known run into hundreds, and most of these happen to be alpha amino acids. These occur either as free or in the combined form, because we know that the amino acids exist as peptides, proteins, coenzymes, hormones, etc. So, in the wide variety of combined forms, they are available in the cell. Further, out of this whole lot of hundreds of amino acids, there are 20 of these are the ones which are fundamental to the sustenance of the life forms, because they are the ones which are the core of the amino acids which we really want to focus attention on to. So, in this whole series of lectures, we will primarily focus on to these 20 alpha amino acids. We will talk more about it as we proceed. Now, if you look at, just for sake of it, the general structure of alpha amino acid, because we know that alpha amino acid, as the name suggests, is an amino acid, that means a bifunctional molecule. The bifunctional molecule is such that is, this amino group happens to be attached to the carbon atom, which is alpha to the carboxylic group. So, that is how it looks like. That means we have a carboxylic acid. So, the carbon atom next to this, say we look at it, this is my functional group here. So, typical IUPAC nomenclature. So, we have a functional group, the carbon atom next to that is alpha, the still next is beta, still next is gamma. So, in case of alpha amino acids, what we have is, we have an amino substituent at the carbon atom, which is alpha to the carboxylic group. That is why the name alpha amino acids. Further, because if you look at this carbon atom here, this is attached to 4 different groups there. So, this is carboxylic acid group, this is NH2, this is just a simple hydrogen atom, and this is R. R basically represents what is called as a side chain, because it is R which differentiates all these 20 different amino acids, because for all of them this part of it will remain the same, whereas this R will keep changing. By changing R, we go from one amino acid to another one. So, basically all amino acids are differentiated on the basis of the side chain in them. We will talk more about it as we proceed a little further. Okay. Now, as regards the nomenclature, how do we name them? Typically, traditionally speaking, the names of amino acids have been given by their discoverers, and primarily they base the names on their sources. For example, the glycine. Glycine has been so named on the basis of the fact that it happens to be sweet in taste. In Greek, glykos means sweet. So that is how the name was derived from here. Another example is, say, tyrosine. Tyrosine picks up its name from its source, casein. Casein basically is the milk protein, which is what is found in cheese. So, tyros again in Greek refers to cheese. So, since tyrosine was found in casein, which happens to be the major protein in cheese, so that is how the name was given to be tyrosine. Further, tryptophan, it basically is the one which derives its name from its source again, that is trypsin, which is an enzyme, and in Greek, phenin means to appear. That means this particular amino acid appears in the enzyme called as trypsin. Similarly, we have got lysine, which derives its name from a tendency to participate in the catalysis part of it, primarily the lysis, that is in German, lysine means breaking down, lysis, and so on and so forth. So that is how the traditional names have been given, but nowadays even IUPAC accepts these traditional names, because that is how do we go about it, but also for all these amino acids we have got formal names in terms of IUPAC nomenclature. Okay. Now, so there are 20 amino acids, as I mentioned earlier. They are the ones which are the most crucial ones. We will talk more about it. So, it is just for sake of it, I have given a table here which gives you the names of all these 20 amino acids. We do not have to bother much about it, because we will come across these very often there. Okay. Now, out of these 20 amino acids, cysteine is the one which is unique. I just want to make a brief mention here about it, because this has a role to play in the structure of proteins. And the uniqueness here is basically this is only one amino acid in which the side chain contains a sulfahydryl group, that is SH group. And this SH group is such that it gives a very unique property to this amino acid cysteine, that is, this can undergo a reaction of this kind, whereas in the presence of mild oxidizing agent, so two molecules of cysteine can undergo a covalent linkage. Maybe these two SH groups here, SH group here, SH group here, they undergo a covalent linkage and give you a very significant disulfide bond. So, this is a very pertinent feature of this particular amino acid only. So, as I just mentioned, that this disulfide bond formation has a very significant role in imparting the structure to the protein, because there are a lot of proteins which rely heavily on the formation of the disulfide bonds on their overall shape and structure. We will talk more about it maybe in future lectures there, where we talk in details about the protein shapes. Okay. Let us move a little further, and then besides these 20 amino acids, there are some of them which again are found in the cell. They take part in a lot of biochemical processes, but and they are very common also, but they are not the ones which are genetically coded. We will talk about it a little while from now. But this some other alpha amino acids, they are the ones say ornithine and citrulline, they are very crucial in terms of metabolism. Maybe at some stage in biochemistry we will learn about metabolic pathways. We find that this ornithine and citrulline are very crucial molecules there, which basically are kind of amino acids. So, ornithine is something which is similar to lysine. It has got one CH2 group shorter in the side chain here, whereas citrulline actually happens to be a derivative of ornithine. Moving further, the another example is, say, thyroxine and triiodothyronine. These two are very important hormones which are found in the thyroid gland, and they play very important role in the body there. Yet another example is that of 5-hydroxylysine, which actually is found in collagen. Collagen is again a very important protein, which is a structural protein. So, that is the kind of structure of this. This basically is a small derivative, that means at 5 position of lysine, 5 carbon here. So, we have a hydroxyl group there. So, it is 5-hydroxy lysine. So, there is some, just for sake of it I mentioned a few amino acids which are very common, but they are not the ones the core of 20. Okay. Now, how do we represent them? How do we represent alpha amino acids? There are two ways of doing it, because typically the amino acids have got names which are very large. If you want to talk about peptides or proteins, if you have to write the names or the sequence of proteins and all, it becomes very tiring or cumbersome to write them. So, what we do is there is some kind of a notations have been given to them, and there are two kinds of notations. The first one happens to be what is called as 3-letter symbols, and the second one is 1-letter symbols. They are the ones we will talk in little details about both of them. Let us start with the 3-letter symbols. The 3-letter symbols basically it is derived from the trivial name of the amino acid, and what we do is we pick up the first 3 letters of the same. That means, we take the common name and take the first 3 letters, and that taken in such a fashion, that is, the first letter is taken as capital and the other 2 are taken to be of lowercase letters. For example, if I want to talk about glutamic acid, now glutamic acid will be represented as GLU, G capital, L and U small. If you write GLU like this, all caps, it is incorrect. If you write all smalls, it again is incorrect. So, 3-letter symbol basically is the first 3 letters of the common name, and first letter of the 3 will be the capital one. Now, when you write, say if I write GLU, basically I am writing glutamic acid, I am representing glutamic acid. When I am representing glutamic acid, I am representing it in terms of a unsubstituted amino acid, and they basically, as I mentioned earlier, are used to save space while representing peptides and all. Okay. Now, let us look at the 3-letter symbols here. What you find is most of it is very obvious. Say alanine. So, first 3 letters are A, L, A. So, we have symbol A capital, L small, A small. Similarly, arginine, aspartic acid, and so on and so forth, because most of them are very straightforward. Out of 20, 16 are very straightforward. But the question comes up is when we talk about, say, asparagine. Now, asparagine cannot be given a symbol as ASP, because ASP has been given to aspartic acid. So, aspartic acid and asparagine, both of them have got the first 3 letters to be common. So, in this case, what has been done is it has been given a special symbol as ASN, N has been picked up from here. Similarly, there is a clash between glutamic acid and glutamine. So, glutamic acid has been given GLU. So, glutamine cannot be given GLU. So, it has been given GLN. So, that is how these two are different here. Thirdly, another clash comes up is when we talk about isoleucine. In case of isoleucine, iso could not be given to this as a name here, because iso actually is a prefix, which is a very common prefix. So, it was found to be leading to some kind of ambiguities at some stage. So, it has been given a special symbol. Start with I, which is given as capital, and then the first 2 letters of leucine part of it has been taken as this. So, ILE is the symbol for this, and rest of them are straightforward. And then this comes tryptophan. Now, for tyrosine, the symbol happens to be TYR, which is very obvious for the first 3 letters, but for tryptophan the symbol could have been TRY. So, there was very likelihood of confusion between TYR and TRY. So, it has been thought apt to give it a special symbol as TRP. So, tryptophan. So, it sounds that way. So, you do not make mistake between tyrosine and tryptophan. So, there are 4 symbols which are, they are not the first 3 letters as defined it to be. So, that is how we have defined the 3-letter symbols. So, that is how do we represent these amino acids, because we do not write the full names always. Now, as regards 1-letter symbols, so this again 1-letter symbol, because what happens is if I use 3-letter symbols, even that also becomes quite cumbersome, because if you look at some peptides and proteins, it may run into say a few hundreds of amino acids. So, if you write the names of 100 of them, so even 3 letters are going to consume a lot of space and time. So, what has been thought of is we give 1-letter symbols to them. So, in 1-letter symbol, each alpha amino acid has been given 1-letter symbol, and that, as the name suggests. So, this letter symbol happens to be the first letter of the common name. So, here again that same issue will come, that there will be more than one amino acid starting with the same alphabet. So, from this point of view, so 11 amino acids, the symbol happens to be the first letter and it happens to be the capital letter there. But for the rest 9 of them, distinct symbol have been assigned, so that there is no ambiguity of any kind. Let us look at the symbols, but before that let me make a statement here. These 3 letter symbols are quite straightforward. We know that the first 3 letters, barring a few exceptions there, but 1 letter symbols are somewhat less obvious, because for 9 of them we have to give special symbols there, and they are not the ones which are coming from the roots of the name here. Let us look at the symbols. So, they are the symbols for this. So, we find that for 11 of them, these blue ones here are very straightforward, but the red ones here are the ones which have been given specially for them. Say for arginine, so it could not have been given A, because A has been given to alanine. So, arginine, it has been given a symbol as R, because as it sounds arginine. So, that is how it has been given symbol as R. That is one way of looking at it. Say for aspartic acid, again because there are 4 of them starting with A, so we could not give A to 4 of them. So, these 3 have been given special symbols there. Aspartic acid has been given D, asparagine has been given N, and so on and so forth. Glutamic acid has been given E, glutamine has been given Q, because G has been given to glycine, so on and so forth. Say phenylalanine, so as the name sounds phenylalanine, so F has been given to this. So, that is some kind of a way of looking at it, but this set of symbols, 1 letter symbols, are accepted and then used globally. So, there is no ambiguity on that. So, if I want to write a peptide or a protein, I can use these symbols to write the sequence of them. So, that we will be talking about in the later lectures there. Okay. So, that is how do we represent amino acids. Now, so what we have done so far is we have just seen what are amino acids, how do we what is their structure and how do we represent them. I have talked a little about their nomenclature. Now, let us move on to the classification. How do we classify alpha amino acids? There are 20 amino acids, and we know that they have got a very diverse kind of structures and all, and we find that there is no unique way of classifying alpha amino acids. There are a wide variety of them. There are different bases. The first basis is the genetic basis. We will talk a little while from now. The second is the nature of side chain. The third one happens to be the dietary source. Now, let us start with the classification of alpha amino acids on genetic basis. On genetic basis, there basically are 2 classes. The first one happens to be coded or primary protein amino acids. As the name suggests, coded, what it means is, so these are the ones, the amino acids for which there is a code, the genetic code is available there. And secondly, these are the ones which are incorporated one by one in the protein as per the instructions contained in the gene for that particular protein over there. That means, I mentioned earlier, there are large number of amino acids are there. We talked about 20 amino acids, then there are some other amino acids, and there are many more of them. So, if I want to just classify them, so there are 20 amino acids which are coded, because they are the ones which have got genetic code. So, from the DNA they will be translated to the proteins there. So, they will be incorporated in the ribosomes and things like that. Now, so they are also called as proteinogenic amino acids. That means, they are the ones which will be forming proteins. The second class here happens to be, as is obvious, which are non-coded or non-proteinogenic amino acids. So, these amino acids are present in the cell, but are incapable of participating in the protein synthesis in the ribosomes. So, coded amino acids are the only ones which will be becoming part of proteins when they are being formed in the ribosomes there. Now, the 20 coded amino acids are the ones which I just mentioned earlier. So, the same list holds true here also. Nowadays, recently in past few years, 2 more alpha amino acids by the name selenocysteine and pyrrolysine have been found to be existing in some certain few proteins there, and these also are found to have genetic codes. Basically, they also are in a way the coded amino acids, that is, and the codes happen to be UGA and UAG. But these are the amino acids which do not get incorporated into the proteins in the usual way as those 20 coded amino acids come into being. So, they are the new discoveries which has been found recently. Cysteine basically is found in number of enzymes, for example, glutathione peroxidase, whereas pyrrolysine is found to be present in certain microbes that produce methane. This is just for the sake of information, that is, though we talk about 20 amino acids, but we know that there are 2 more added to the list, but there more information is coming on these amino acids. Okay. Now, let us move to the next basis of classification, that is the nature of side chain. So, on the basis of nature of side chain, there are 3 different classes, or you can say 3 groups are there. The first happens to be non-polar or apolar amino acids. Second happens to be neutral or uncharged polar amino acids, and the third one happens to be charged polar amino acids. Let us look at each one of them. Now, non-polar or apolar alpha amino acid, it is very obvious. That means the side chain happens to be non-polar. That means primarily there will be aliphatic side chains there. So, look at glycine is one exception, because in the side chain we have got only hydrogen atom, because glycine is exception in more than one ways. We will talk about it as we move further. Right here, so glycine is one amino acid for which the side chain happens to be just a hydrogen atom, so it is non-polar. Alanine has got a CH3 group, say valine has got isopropyl, and so on and so forth. So, glycine, alanine, valine, leucine, isoleucine and proline, so all of these belong to because there are aliphatic side chains are there. Proline also belongs to this group here, because it does not have polarity in the side chain there. Proline also has got one more kind of uniqueness to itself, because this is only one amino acid which is not an amino acid in true sense of it, because in this case what happens is, if you look at, generally we got NH2 here, this is my carbon atom here, this is CH alpha, this is my COH, and this is my side chain here, so this is alanine here. So, this is my common thread in all amino acids there, that means NH2, CH alpha and then COH group. So, in this case what happens is, in case of proline, the amino group here, this it gets into, that means there is linked with the side chain here, that means because otherwise this amino CH and COH has to be there, but this amino group is a part of the side chain as well. So, now this actually becomes a imino group. So, if you really ask me, one should call proline as a amino acid rather than alpha amino acid, but it is collectively used in the same way as other amino acids there. There are 4 more amino acids which belong to this class. They happen to be methionine, cysteine, phenylalanine and tryptophan. Here again, so this is SH group here, but it still is non-polar. So, that the prime thing here is that they are non-polar, because it has a very significant implication in terms of structure, because these side chains which are non-polar or aliphatic in nature, they will be hydrophobic. So, when the protein structure comes into picture, how does the protein fold, what kind of shape does it require. So, these interactions will become very pertinent over there. So, these amino acids have a role to play, all amino acids have a role to play in terms of their determination of the structure of proteins. We will talk more about it as we move further. The second class is neutral or uncharged polar. That means, as the name suggests, the side chain is neutral, there is no charge separation, but still there is small amount of polarity in that. So, there is some kind of a polarity coming into picture. For example, say tyrosine, it has got, so this is my CH2, a benzene ring here, and then this is OH, it is basically phenol part of it here. So, this is phenolic group OH. Threonine, this is my OH again, serine also there is OH, and so on and so forth. So, we find that these amino acids, the side chains are not charged, but they have got some kind of a small polarity in that because of the functionalities available in those side chains there. The third group is charged polar. That means, now the side chain has a charge, and of course, if it has a charge, it will be polar. The example happens to be histidine, arginine, lysine, aspartic acid, glutamic acid, the 5 of them. Lysine is the one, this is my typical backbone here, the side chain there, CH2, 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 the 4 of them, and then there is a NH2 there. So, this is the one which is causing it to be, because NH2 is going to pick up a proton, become NH3 plus, so it will be charged there, it will be polar. So, this basically is an example of a basic amino acid. Even glutamic acid, you see in the side chain we got a CH2, we got a COOH. There is an additional, because besides this NH2, CH, COH, we got a COH group in the side chain also. This is again is a carboxylic acid group there. So, this will be again polar. Okay. So, this is the one way of looking at the amino acid. Probably one classification could be acidic and basic amino acids. So, these two are examples of acidic amino acids. Lysine and ornithine, they happen to be examples of basic amino acids. Even histidine is a basic amino acid. So, is arginine, because the side chain happens to be basic in nature. Okay. So, that is the way we classify amino acids. Okay. Now, having done talked about the classification, let us now move on to yet another way of classifying, that is on the basis of the dietary sources. Let us look at what do we mean by this. We know that amino acids are synthesized in the body. All living organisms, plants, animals, they all synthesize amino acids, but there are certain higher animals like human beings. So, they are not able to synthesize all the amino acids, because our body needs all the amino acids for a variety of functions, because we need a lot of enzymes, we need a lot of proteins, so many of them are required, but our body can synthesize some of the amino acids required for that, but others are not being made in the body. So, we need to have some dietary source for that, and on that basis we can classify amino acids into 3 groups. They happen to be essential alpha amino acids, semi essential alpha amino acids, and non essential alpha amino acids. Let us look at each one of them. The essential amino acids, as the name suggests, so these cannot be synthesized by the body, they must be provided by the dietary source. That means they are essential to be present in the dietary sources. The food we take must have these amino acids. If they are not there, if we are taking food which is devoid of these alpha amino acids, we will be having a lot of implications in terms of health and all. Now, the essential alpha amino acids, they differ from species to species. For human beings, there will be certain group. For other animals, there may be a different set of amino acids which will be essential there. So, their food will need those amino acids. For human beings, there are 8 alpha amino acids which belong to this category. They happen to be leucine, isoleucine, valine, lysine, methionine, phenylalanine, threonine, and tryptophan. So, there are 8 of them. You must have heard about lysine rich bread. Now, a lot of breads come which are rich in lysine there, because that has been added from outside. So, that when we consume that bread, you are taking lysine which is required by the body to prepare proteins in it. Now, the second class is semi essential amino acids. Semi essential, what does it mean? What does it mean is the following: these amino acids, they are the ones which can be synthesized in the body, but they are synthesized at a rate which is not sufficient enough. That means, we need certain amount of amino acids, certain rate is required, they are not formed at that rate. So, it is advisable that these are available in the diet, not necessarily, they are not essential to be present in the diet, but if they are there, they will definitely contribute to the well-being of the organism. And the 2 amino acids which belong to this category are arginine and histidine. Some of the authors even club these 2 amino acids along with the essential amino acids, and then they maintain there are 10 amino acids which are essential. But you must distinguish between the 8 which I mentioned earlier and these 2 which are being synthesized in the body, but at a rate lower than what is required. Now, so they need not necessarily be present in the diet, but if they are there, they definitely are advisable or desirable. The third category is okay. Before I move to the third category, let me make a mention of 2 very important amino acids, cysteine and tyrosine. They are very ambiguous in terms of the classification as we are talking about, because these can be synthesized in the body, there is no problem on that, but they consume, that means cysteine to be made, that uses methionine, but tyrosine uses phenylalanine That means we need phenylalanine to prepare tyrosine, and these two amino acids, methionine and phenylalanine, happen to be essential amino acids. So, the body can make cysteine and tyrosine, but it uses two important essential amino acids for the purpose. So, it is better that if these are present in the food, as this will deplete otherwise the two essential amino acids. Suppose they are present in the diet, it is always good. Otherwise, what will happen is they will consume these two amino acids which are essential, they are not being made in the body, that means we will have to provide from outside. So, if they are there, that is really great. Third class is basically non-essential amino acids. So, they are the ones, the rest of them, that means we are listed about 8 plus 2 plus another 2. So, there are 12. So, the rest of them, that 8 of them happen to be belonging to this category, they are called as non-essential amino acids. What does it mean is that this can be synthesized in the body and need not necessarily be present in the diet, but that does not mean that it should not be there. If they are there, fine. But when I say they are non-essential amino acids, it does not mean that these are not required in the body. Our body needs all 20 amino acids, all are important. The only thing is, when we talk about classification of this kind, essentially we are referring to the dietary sources. I mean, they are not essential in terms of their being present in the diet. Otherwise, they are essential for the well-being of the body there. Let us now sum up what we have done today. What we have done today is, we started by putting the topic, that is amino acids, in perspective. That means, where do they fall in our whole scheme of things. Thereafter, we saw what are amino acids, what is their importance, and then how do we name them. We talked about the nomenclature of alpha amino acids in terms of the traditional way of doing it, wherein we said that they are named on the basis of the sources and some of the physical properties. Thereafter, we took up the representation of amino acids. In the context of representation, we talked about two ways of representing them. One is in terms of 3-letter symbols or 3-letter codes, and 1-letter symbols. We saw that what is how do we represent them, and what are the rules for representing them in this kind of symbols and all. Thereafter, we had a detailed account of the classification of alpha amino acids on different bases. We started with the classification on the basis of genetic basis. On that basis, we said there are coded and non-coded amino acids, and thereafter we talked about classification on the basis of the nature of side chain, where we talked about the amino acid side chains to be say apolar or non-polar, and then uncharged polar, and then polar, and so on and so forth. And then each one of them has a significance in terms of, we did highlight this fact that they have significance in terms of the overall structure the protein is going to adopt at a later stage. Thereafter, towards the end, we took up the classification of alpha amino acids in terms of their dietary source, and in that context we defined 3 different classes, that is essential amino acids, which are essential to be present in the dietary source, and then there are semi-essential, they are desirable to be there, and non-essential need not be there in the diet, but we did conclude by saying that all alpha amino acids are essential for the body. It is just that only in the context of their being present or not present in the dietary source, we put them into different classes there.